Hey everyone, we're going to go over DNA structure today, and I'm going to start with this image. And so this whole thing here is DNA. And so let's look at the parts of that. The D stands for deoxyribose. If you see ribose, that usually means sugar, or actually if you see OSE, that usually means a sugar. And so we can see all those sugars here in orange. Next we have Na, which stands for nucleic acid. And so the reason it is a nucleic acid is because it has these. Those are nitrogen bases. And so let's go over the parts of DNA. DNA is, again, a nucleic acid. A nucleic acid is a polymer because it has multiple parts. So if nucleic acid is a polymer, that means there is a monomer. The monomers have 3 parts to them. Each of those monomers is a nucleotide. Let's look at those monomers. This monomer, for example, has a sugar, which is deoxyribose. If we look at this sugar, it consists of lines. Each of these changes in angle represents a carbon. And so you can see there are 4 carbons here. However, there's actually 6 carbons. And that is because, as you might have noticed here, there's another carbon right there. Carbons are so frequent in chemistry that we don't even bother drawing them really. And if we were to count all these, it'd be 1, 2, 3, 4, 5. I'm sorry, I meant 5. If you remember, carbohydrates come in a 1 to 2 to 1 ratio. So that tells you there must be 10 hydrogens and 5 oxygens. And so because we can number these, there is an end to this. There are 2 ends. There's a 5' end and a 3' end. And we can see that if we look closely over here. 5' end is this carbon. 3' is this. Let's draw that again. So that means that this sugar is face up. On the 3' carbon, we have a phosphate group, which is just a phosphate surrounded by oxygen. In case you're wondering what this double bond means, it basically means that 2 electrons are being shared rather than just 2 pairs. Next we have the nitrogen base. Off of the 1' carbon. And I'm just going to draw A here. But this is the shape of adenine. Adenine has, if you look carefully, has a lot of nitrogen. And that's true for all of these. That is why we call all this stuff in the middle nitrogen bases. Look at all the nitrogen. And so I will just draw A to represent adenine. This entire structure that I've drawn now represents a single nucleotide. The entire structure here with all of these nucleotides together is nucleic acid. If you look at the words nucleic acid, you see that each word starts with a letter. The first is N, the second is A. And that is why DNA has NA as part of its acronym. And so let's look more at the nucleotide. The part of the nucleotide that is the code is the nitrogen base. We now have 3 different words that start with N, and so it's very easy to mix these up. The smallest unit is the nitrogen base. The second biggest is the nucleotide, and then the biggest is the nucleic acid because it is the polymer, whereas this is the monomer. And so what does the phosphate do? The phosphate connects one nucleotide to the other. And so if I were to draw another nucleotide here, I would simply take the 5' end of another deoxyribose sugar and connect it. And maybe this one is, I don't know, G? I think that's right. No, sorry. C for cytosine. And so the phosphate is what is responsible for connecting this together. That is why we call this part of the DNA the phosphodiester backbone. We call it ester because of this here. Two carbons connected together with hydrogens around them is called ester. It's just a chemical group. No need to remember that. Just remember this, the phosphodiester backbone. The phosphodiester backbone has properties that are important in molecular biology. One of which is that if you look, it has oxygens with negative charge. Therefore, the phosphodiester backbone is said to be negative in charge. This will become apparent later when we use certain molecular techniques to analyze DNA. And so let's look at these nitrogen bases a little bit more closely. If you notice here, A always pairs with T, and C always pairs with G. But if you look really closely, you'll see there's a difference with how they pair. Try to see the difference. Look at those dotted lines. What do those dotted lines represent? Here, here. These dotted lines represent temporary bonds, and we've heard of temporary bonds before. Dipole moments of polarity allow for molecules to temporarily bind to each other in what's known as hydrogen bonding. We represent hydrogen bonding with dotted lines. As you can see, A and T have two hydrogen bonds. Whereas C and G have three. And so C and G is actually a stronger bond than A and T. Each of these hydrogen bonds is singularly weak, however collectively very strong. To get a feeling for how strong hydrogen bonds can be, try getting a 5-gallon bucket. Put just a little bit of water in it. And then stick another 5-gallon bucket in there. Try to pull it out and you'll see just how strong hydrogen bonding is. It's nearly impossible when there's a lot of it. And so though each of these is weak, collectively it is strong. This is important because if DNA needs to be read or edited, we have to separate these two strands. We also separate these two strands when we copy DNA because we have to take one strand, split it in two, and put a new strand on it. And so it's good that each of these is weak because we can easily start unzipping it from one place. However, collectively still very strong. I mentioned earlier that there was a 5' end here and a 3' end here. Just a reminder. If we look at how DNA is made, we see that DNA is parallel. However, the 5' end is the end that is up, and the 3' end is the end that is down. And so although they are parallel, they are actually anti-parallel. We then twist that, and we make a double helix. Okay, so that's the overall structure. Let's talk about how we condense DNA. We condense DNA into packages called chromosomes, and you've seen chromosomes before. You know that you have 23 pairs for a total of 46. You also know that genes are located on these chromosomes. You should also know that chromosomes in this shape are made during S phase. They are duplicated for the sake of division later. Chromosomes in this shape have sister chromatids. Two of these together would be a homologous pair. You get one from your mom, and you get one from your dad. Homologous pairs also have the same genes, just like sisters, but the alleles can be different. Chromosomes are just highly compacted segments of DNA. If you were to start pulling at, say, a gene, you would unravel the DNA, like a strand being pulled from a sweater. Here's the overall structure of that DNA. We are zoomed out here, zoomed in, and we are zoomed out here. On the right, you can see that DNA is a double helix. This is visible only under the greatest of magnifications. We then take this double helix and we start zooming out as we go this way. And you can see that it starts to become indistinguishable as a double helix at this point here. It just looks like a thread at this point because we've zoomed out so much. We then take DNA and we wrap it around these proteins, sort of like how you would wrap a cord around a battery box to save space in your computer laptop case. We take these round proteins and we wrap DNA. These proteins are actually more interesting than they look. They're not just round. They actually have 4 parts, or 8. And each of these units has a tail that comes out, called a histone tail. These tails interact with environmental elements to help determine how condensed DNA is or is not. For more information on that, look at metagenetics. To find out how environmental factors can actually influence gene expression, a new field in biology. These cool proteins, these histones, coil up into units of 8 called nucleosomes, and those nucleosomes then begin to stack to create coils. Coils themselves coil into supercoils. Those supercoils are then knitted together into a complex shape that eventually makes a chromosome. And so you can see that chromosomes are very dense. If you were to unwind a single chromosome, you'd probably find about 1 meter of DNA. For your 23 chromosomes in total, oh sorry, not 1 meter. For 23 chromosomes in total, or 23 pairs, that is about 5 meters of DNA per cell. And you have 100 trillion cells. That's a lot of DNA. Let's look at the terms over here. So we just talked about nucleosomes. Chromatin is this structure here that creates the chromosome. A chromatid is a single unit of a chromosome. Chromatids are made during S phase of the cell cycle and are only there for division. Chromatids have identical genes and alleles, unlike homologs, which may have identical genes but possibly different alleles. Here's the overall structure of a chromosome. The centromere consists of 2 parts. These are called kinetochores. Kinetochores are pulled on by centrioles during cell division. These centrioles use microtubules to pull against the chromosomes, thereby dividing the kinetochores and splitting the sisters. This counts as 1 chromosome, as does this. However, this chromosome here has 2 sisters which are identical. This ensures that a new cell made by splitting this will be identical to the old. I'm just repeating myself here. I'm going to move on. This is the human karyotype, consisting of 23 chromosomes, and you can see that the last pair, as we've talked about before, determines the gender. You can also tell another thing by looking at this. If you look at the 21st chromosome, you can see that this is a normal individual. In some individuals, you have a 3rd chromosome, and that is known as trisomy. Tri means 3. Trisomy results in Down syndrome. Down syndrome results from having 3 pairs of chromosome number 21, or 3 individual chromosomes of chromosome number 21. That is caused by a defect in meiosis. And so basically, we've learned before that in meiosis, tetrads form from homologs where crossing over occurs. Usually, these would be separated and pulled apart. However, sometimes they are not pulled apart. This process is known as non-disjuncture. Non-disjuncture. Let's watch a quick video on genes, DNA, and chromosomes. Or maybe not. How do I watch this? There we go.
You can think of DNA, genes, and chromosomes as forming a kind of blueprint of life. These are the plans that tell the body how to make things, in this case proteins, which are the body's basic building blocks. But unlike a blueprint for a house where all the information is stored on a flat piece of paper, the blueprint of life has many levels of organization to it. Let's start with DNA, the most basic level. A DNA molecule is made up of a sugar backbone and 4 flavors of a molecule called a nucleotide. These nucleotides are arranged along the backbone in a unique order. When your body needs a particular protein, it reads the sequence of nucleotides and uses it as a template to make that protein. Moving up a level of organization, we have genes. Genes are long snippets or chunks of DNA. They're also the most basic unit of inheritance. Traits are passed on from parents to children in the form of genes. Genes include other information about building proteins. For example, they tell the body how long a protein should be and how much to make and when to make it. Genes also have directions about where DNA can be edited out to make proteins with different functions. This is how the body makes an estimated 1 million proteins from only 20,000 genes. The next level up is the chromosome. Humans have 46 of these, arranged in pairs. Chromosomes are long strands of DNA wrapped around beach ball-like proteins called histones. Chromosomes contain many genes, along with long stretches of DNA, whose purpose we've yet to figure out. In order to make a protein, DNA has to be unwound from its histones. This is controlled by a process called acetylation, and it's just another level of control the body has over the production of proteins. So as you can see, the blueprint for our bodies is quite well organized. If only that was the case for the rest of our lives. For Scientific Americans.
Right, so he mentioned proteins in there, and that is true. We are going to learn pretty soon how proteins help to proteins are created from genes. All right, so that's pretty much all today. We have talked about nitrogen bases, and those are here. These are the different nitrogen bases. Genes. Genes are a sequence that codes for a protein. So, for example, the melanin gene might make the melanin protein. I'm trying to think of another one. The lactase gene makes lactase protein. An allele is just a version of a gene, so it's usually a dominant or a recessive version. Chromosomes are highly knitted, condensed forms of DNA. A sister chromatid is an identical copy of DNA. And then a homologue is 2 chromosomes with different alleles. And you get 1 from each parent. All right, that's all. Till next time.